My name is Chen Su, and I'm a graduate student at McGill, working under the supervision of Dr. Amin Ahmad. Today, I will talk about the multi-modal inference of phenotype-relevant transcriptional regulatory networks in embryonic development using InferNo-ChIP. Even though the gestational time of a human is around 9 months, most tissues and cell types are specified in the first few weeks post-conception in the course of human embryonic development. After fertilization, the resultant zygote begins to divide and then forms a blastocyst. After the implantation of blastocyst, the innermost layer of it will form 3 germ layers: ectoderm, endoderm, and mesoderm. Those germ layers then give rise to different cells. The formation of those cells described here is driven by the activity of transcriptional regulatory networks, or TRN for short. A TRN is a complex network of regulatory relationships of TFs and genes, where TFs are proteins that can bind to specific DNA sequences to control gene expression. Reconstructing such networks helps us to better understand the regulatory mechanisms involved in the course of human embryogenesis, which allows us to understand common diseases in pregnancy and birth. Many methods have been developed to reconstruct TRN, but most of them do not relate TRNs to the variation in the phenotype. So what do I mean by phenotype-relevant TRNs? For example, consider an experiment that involves 2 conditions, case and control. Both figures here show a linear correlation between expression of a TF and a gene across all samples, which can then be interpreted as the evidence of regulation. But the relationship in the figure on the left is likely not to be relevant to the phenotype, while the TF-gene relationship on the right is likely to be phenotype relevant. Our lab previously developed InferNo to incorporate gene expression and phenotypic labels of scores. In this study, our goal was to generalize that model to enable incorporations of ChIP-seq data in order to study differentiations of human embryonic stem cells to endodermal lineage. As inputs to our new model, InPheRNo-ChIP, we used RNA-seq data from 3 independent studies, ChIP-seq data, as well as phenotypic labels. InFeRNo-ChIP utilized a probabilistic graphical model, PGM, to integrate regulatory evidence between TFs and genes with phenotype relevance information of genes to reconstruct a phenotype relevant TRN. Here, I'm showing you the pipeline of InFeRNo-ChIP. In step 1, we perform differential expression analysis on the integrated gene expression dataset to obtain p-values associated with how much a gene is differentially expressed in human embryonic stem cells versus endodermal cells. Then, in step 2 and 3, we obtain 2 lists of p-values of TF gene associations from ChIP-seq and RNA-seq data. Those p-values are then fed into step 4 of InFeRNo-ChIP, which is a carefully designed PGM to model the conditional distribution of those summary statistics and generate an initial bipartite graph. As the last step, we perform several normalization steps and comparison with different controls to identify TF gene relationships that are associated with the differentiation process. The PGM in the step 4 is the core of InFeRNo, which I will describe in details. The goal of the PGM is to model the biological hypothesis about how each element influences each other. To design a PGM, we first define a binary random variable Tij for each path in the bipartite graph. Intuitively, if Tij equals to 1, it means TFI is regulating gene J, and gene J's expression is associated with variations in the phenotypic labels. On the other hand, if Tij equals to 0, it means at least one of those 2 statements is incorrect. Therefore, the TRN reconstruction problem can be formulated as obtaining a posterior probability on each Tij. Then, we carefully describe the conditional distributions of our observed p-values and latent variables, If you are interested in its details, please stop by the poster and I would be happy to expand it. As the first evaluation, we compare the TF gene ages identified by our algorithm with the GTRD database that provides an aggregated TRN based on the integration of many different studies. While this aggregated TRN is not phenotype relevant, we expect to see an enrichment with regards to ages we have identified, which we'll observe with p-values of 2 to the power of -12. After we identify the TRN, we rank TFs based on the numbers of target genes in the TRN. Such TFs are expected to be important in the course of endoderm formation. Consistent with our expectations, we found various literature evidence for the role of those TFs. We used the experimental study as our primary data to further validate TF gene phenotype relationships identified in the TRN. This study uses RNA-seq and ATAC-seq data to investigate dynamics in chromatin and sensibilities during the differentiation of embryonic stem cells to endoderm. Then they perform perturb-seq screening to define the loss of function phenotype of potential molecular drivers in endoderm development. 3 TFs in our study were shared with this study. Focusing on those TFs, we found that in all cases, we observe a significant enrichment in the target genes identified by InPheRNo and the target genes experimentally identified by this study, which is associated with early endoderm development. This finding confirms the ability of InPheRNo-ChIP to identify phenotype-relevant TRN. Thank you, and I look forward to seeing you in the poster session.